So I'm going to do that. I think I'm actually going to also drag you through. If I'm going to record this, I'm worried what it does. So as far as everybody knows, the video notes are all right. Is that correct? If nobody, I've had people point out to me when they're wrong. I'm going to delete this one and this one. My computer doesn't have enough room for all this stuff. There we go. All right, so hopefully that'll make sure we make it through. All right, so to get the help session, that's on Google Meet. We have lab reports are due next week online. Just around the website. On Tuesday at 11:59 p.m. Okay, so we're not confused about which midnight we're talking about there. You could ask questions in the lab if you needed to about those. Okay. Probably not between the labs. I kind of need that time to recover. Let's see, what else? We're going to do lablet next week. And I think that's everything. All your lab data is up on the website, so what I'd encourage you to do is go look at it, try to make sure you can access it right now. In terms of the spectra you're going to plot, you've been given a Excel file with just the data, so you actually have to make the plots with those. I don't care how you do it, so if it turns out you're pretty good with AI and you can do your AI plots with them, sure. AI doesn't usually mess up plots. You saw them before, so you have an idea what they look like as well. What I want you to do as well with those Excel files is, in terms of opening them or seeing if you can open them, you're going to need to download them and then click on them. Maybe you'll be lucky and you won't need to do that. You'll just click on them and they'll open up. But I have had people in the past who have problems where they click on them and it doesn't work. So download them, click on them, make sure you can open them, and make sure you've got a mess of data, wavelengths and absorbances. Okay? And I told multiple of you during lab that the saving process is complicated, and so I'm doing it rather than giving it to you guys, but I also make mistakes in it. There's just too many little things that have to be done between what you got in an Excel file. Questions? Just check it, make sure all of it's there. You can tell me on Tuesday it's not there, but it won't give you a lot of time to do anything about it, right? Yeah.
The photos are very grainy.
What?
The photos are really grainy. You can't really see anything.
Well, yours might. I can't. You'll have to show me later. It might be that when I took your photos, it wasn't in focus. I can see things when I look at that from here, though. So come talk to me, and we can talk about it. But work with the photos you have. I can't make new photos. But come talk to me if you're concerned about that. Any other questions? All right, then we'll keep going on. So we've been talking about, last lecture, we talked about pKa's at the end of lecture, right? So we have this magic thing called a pKa, right? It's the negative log of the Ka. And so the pK, when the pK equals the pH, what does that tell us?
The concentration of the acid is equal to the concentration of its conjugate.
Yeah, the concentration of the acid and the concentration of the conjugate base are equal. Okay, so that's really an important thing that we're going to use in solving problems quickly. So when we look at that, and that falls out of the Henderson-Hasselbalch equation, which, remember, is pH is equal to pKa plus the log of A minus, so the conjugate base, over the concentration of the acid. So I've written that like that. Those are molar, right? When I put the brackets around them, they're molar. But for this equation, does it matter whether I have them in molar or not? You're always safe if you put them in molar, but if you get them in some other units, are you okay here? And if yeah, if so, because that's the way I'm asking it, right? Why? Yeah, the units cancel out. Excellent. Okay, so if this in turn was not molar and it was millimolar and millimolar, as long as they're both the same units, the units cancel out and you're just looking at a ratio of the two, right? So when we talk about the acid and conjugate base are equal, well, they might both be 1 millimolar. That would be crappy buffer. We talk about buffers later, okay? It could be 100 millimolar, but they're both the same. And we would get the same pH out of that, okay? So we get the same pH regardless of the absolute concentrations of these, as long as we have the same ratio. And when these are equal, 5 over 5, I get a 1, log of 1 is 0, this term goes away, those two are equal. Does that make sense? Why? With the math? All right, so this is something very useful for us, that we can quickly look at the pH and the pK and determine whether a functional group is protonated or not, okay? Whether it's protonated or not, and not whether it's fully protonated, right? These are all, it's partially protonated, partially not, right? At 50, when these are equal, you got half and half. It's half protonated and half not. If you have a little bit, if I have half protonated and half not, and then I take the pH and I lower the pH a little bit, what happens? I had, these were equal, I lowered my pH, what happens? What was that?
They're not equal anymore?
They're not equal. That's that partial credit.
The reaction goes towards the conjugate base.
The reaction is going to go give us more acid,
Oh.
right? We lower the pH, we have more protons around. I'll draw out the Le Chatelier's principle here in a second, right? But we have more protons around. But there's two ways we can think about this, okay? We can think about it using Le Chatelier's principle, which, you should apply Le Chatelier's principle wherever you can, as often as you can. Or you could think about it, if I lower the pH, I'm putting more protons in, right? Got more protons, they stick to things. Okay, if I raise the pH, I'm taking protons out, right? Higher pH, fewer protons, and they come off of things. Take protons off, they come off of things. But let's go ahead and look at what this reaction is. So we could also use the equation. I don't feel like that's very intuitive with the equation. So if I've got my acid base, and this is the Ka, so it's the acid dissociation reaction, like that. If I lower my pH, that means I'm doing what with this H+? I'm making more of it, right? It's confusing. Lower means more. And then the reaction due to Le Chatelier's principle will go off to the left. We'll get more HA. That makes sense? Yeah.
In like a practical setting, how would we do this? Adding different acid?
So if you added a different acid, right? If you added a different acid, you'd get more H, right? If you added this acid, you're gonna, whatever this acid is, you're gonna push it that way. Okay. There are others. So we can think about this from the perspective of buffers. We're thinking about here just as kind of an abstract HA, but what I'm really concerned with is it's going to be like parts of proteins. And we're going to ask this piece of the protein, this functional group, what happens to it if it becomes more acidic? That's really where it's practical. And even more practical, so I'm asking, you could just add a tiny bit of acid here when these are equal, right? And suddenly it's more protonated than it isn't. And if you were doing, so my research is this simulation chemistry, right? I'm doing molecular modeling. I have proteins that are running around the computer. And to make things simple, if you just have a tiny more, if the pKa is just a tiny bit lower, or the pH is a tiny bit lower than the pKa, means you have a tiny bit more of this than that, you say, okay, you got that. Because you can't do all of it and have ratios of every functional group when you do the calculations. If that makes sense. It bothers me, right? But that's a limitation we have when we do that stuff. All right, so let's go ahead and take a look at this, and let's look at some interesting functional groups, or the main functional groups we're going to talk about in biochemistry. We'll talk about these more than anything else. First of all, we have a carboxylic acid. And I talked about this before, but I'm going to talk about it again because it's important. We've got a carboxylic acid, and that can go to make the carboxylate, so the conjugate base, right, plus a proton. Okay, the pKa of a carboxylic acid is about 4. Not always. Okay, depends on what's around it. Will influence it. Depends on what functional groups are on it that are near it, and what things are near it in three-dimensional space. All right, and then the other functional group that's going to be really important to us is going to be amines. And so the amine can deprotonate, or the ammonium, I don't know what to call it exactly when it's on, it's attached, it's ammonium-like thing. And then that can deprotonate. And that pKa is about 10. These are the most important functional groups in biochemistry. Go do research, you might find other ones that are important, right? But broadly in biochemistry, those are the most important functional groups. I expect you to memorize that it's about 4 and it's about 10. Okay, so I could ask you, what's the protonation state of this? And if it's an amine or a carboxylic acid, I don't need to tell you the pKa, as long as I'm not being subtle with where the pH is. All right, so you've written that in your notes. I'm going to scroll down, or actually, maybe I'll just write it over here. Let's do a quick problem here. What is the major species for carboxylic acids and amines at pH 7, right? pH 7 is the main pH. We're going to talk about pH 7 a lot. If we want to be more specific and talk about blood, we'll talk about pH 7.4. But what's the major species for each of those? You can circle it in your notes. Anybody think they've got it? Lorenzo? Okay, is it the neutral species for each one? I'm thinking not. Yeah.
The carboxylate
Claudia.
and then
The carboxylate and then the
amine on the left.
and the positively charged amine, the protonated form. Right, right. So our major species are going to be, this is going to be major at pH 7, and this is going to be major at pH 7. Okay? And so the way to reason that is, if we look here, my pK is about 4. 7 is higher, right? So more basic. So there are fewer protons around, right? So then I could go ahead and I could grab this and pull it down. There are less of those. We go this way. That's my major species. Does that make sense, the reasoning? Okay, down here, the pK is 10. 7 is lower than 10, more acidic, more protons. So we'll go ahead and add the protons and move it that way. Okay? These are your major functional groups that we're going to be putting protons on and off of. And what I want to remind you is that their nitrogens are going to be, I said this earlier, yesterday, nitrogens are going to be positive or neutral, right? And oxygens are going to be neutral or negative, always. Okay, so despite my making a hydronium ion, All right. That happens because water is so concentrated. I didn't have a good answer at the time, but I wanted to look up what the pK of water is, and I did. And pK of water is 0 or less. Okay, so it's super. It's very acidic. It's a pretty strong acid. The pKs of your really strong acids are even lower or negative. Okay, so that's why you could throw hydrochloric acid into water and the protons all come off. Questions on that? How you would do that? Okay, I was going to give you. I was trying to look up what kidney tubule pH was, and this is the AI. This is the AI answer that was at the top of the page. Go ahead and read through it and see if you are happy with it. And you can go ahead and write down what you think. What do you think is wrong with this? Yeah, Sophia, what do you think's wrong?
Wouldn't it be like below 5.9?
Yeah, yeah. So we can read through this. Let's say I trust the AI where it says normal healthy kidney will typically produce urine with a pH of 4.5 to 5 to acidify the body. Okay, I'm not sure I agree with that either, right? That's less acidic than your blood is. So you're not really acidic. You might be removing acid from the body if you do that, but that's okay. Let's accept that. Let's ignore that sentence. Let's accept that that's correct. And then it says when somebody has renal tubular acidosis, the urine pH remains above 5.5. Really? It's renal tubular acidosis. That seems wrong to me. Okay? All right. So anyway, don't trust the AI answers. All right. Not trying to get you to not use AI, but be careful with it. Yes.
That does seem weird, but is it saying that like it's the urine is above 5.5 and it's not able to get rid of the acidity? Like it's, I don't know.
Let's take a look at that.
I don't know if I'm thinking of that the wrong way, but like.
So it's saying the urine pH remains above 5.5 despite the body's need to excrete acid. Okay. I see that. I see what you're saying there. So it might be that's what's going on. All right. But if that's the case, I definitely still have an issue with we're not acidifying the body by excreting 4.5 to 5.0 because your body, your blood, which is what interacts with the kidneys, right, is pH 7.4. So that's a good point. But in any case, be careful with these sorts of things. I would definitely check any of these out to make sure that they are correct. Sometimes the AIs are getting better all the time, right? Maybe, but in terms of AI, when I do my research, which is programming, I'm using it like all the time to do the programming. And then I'm checking it, right?
You should ask me a follow-up question.
What was that?
You should follow up with it.
Ask it about that.
Yeah.
I've had AI run me around in circles where I say you're wrong, and it'll apologize and give me another answer that's also wrong when it doesn't know, right? There's some things it's really good at, but sometimes it doesn't have things right. Okay, I've got DNA molecules up, but we're not doing that yet. Let's go ahead and do a practice problem. Now we're using a Henderson-Hasselbalch equation. So Tylenol is it's a drug used to treat fevers. Treat fevers and pain. Certainly gave me a lot of Tylenol for my pain. It also causes liver damage in a fairly large fraction. There's an unacceptable fraction of people at its maximum dose.
What is the unacceptably large fraction?
I don't remember. I have 30% in my head, though.
More than 2%?
No, I have like 30% in my head. So, but it may not be that may not be the correct one, but it does. And what actually where people really get into trouble is if you're taking your Tylenol and then you've got something Tylenol acetaminophen and you also have acetaminophen in other things you're taking. So it's not a place where you've got a lot of tolerance above that max dose. So you end up with a lot of people end up with liver damage either by being at that maximum dose for a long time or they're taking multiple things that contain acetaminophen. Okay, so it's actually a pretty dangerous thing. And if it was to try, they were to try to pass it through the FDA today, it wouldn't pass. Here's the structure of Tylenol. That said, if you had a fever, if you had a really bad fever, and you had to interleave your ibuprofen with your Tylenol, I would do it. When you can't get the fever down just with the ibuprofen alone. I would do that. And I'm not actually supposed to take ibuprofen because it inhibits bone growth to some extent, and my back is still fusing. And so I will take Tylenol instead of ibuprofen. But I'm really careful not to be taking it at the max dose for a long time. Here's the structure of ibuprofen. Not ibuprofen, I'm sorry. This is the structure of Tylenol. Also want to be, there are plenty of things that you might take that they wouldn't know the full mechanism for. How Tylenol works, how people think Tylenol works changes about every 5 years, which is a little bit concerning too. Ibuprofen, aspirin, they know exactly how that works. They know what enzymes to target, but Tylenol, no. Okay, so here's the structure of Tylenol. I was going to ask you which proton would come off, right? But how would you know which proton is coming off? How do I know it's this one and not that one? Claudia?
Oh, because the oxygen is negatively charged.
Because the oxygen would be negatively charged. What would happen if I took a proton off of this nitrogen? It'd be negatively charged, and they're never going to be negative in here. Okay? All right? It's in a mid. Amids, the proton doesn't come off. We're going to see a lot of amids in here. They're peptide bonds, and those are never going to come off. Don't do it. Never take it off, and never put another one there either. Never two. Never make this positive here. It is not going to happen between pH 0 and 14. Okay, so let's go ahead and ask a question though here. So I already drew which proton comes off, but the pKa of this is going to be 9.5. And so then I could ask a question. What is the percent abundance of all forms of Tylenol? Actually, let me use acetaminophen. acetaminophen at, let's say, pH 8. All right, so this is a Henderson-Hasselbalch equation. We would set that up. We're going to go pH is equal to pKa plus the log of A minus over HA. All right, which of these is my HA? The one on the left, yes, exactly. So I have HA up here, and I have A minus here. All right, when you do these problems, it's going to be important that you indicate, you can come up with your answers, the percentages, but you need to indicate which structure is which, right? So you can tell me HA is 10%, but I need you to also tell me which of the structures is HA. All right, so we have a pH and we have a pKa. My pH is 8.0. My pKa is 9.5. And then I've got concentrations of my conjugate base and my acid, and I don't know what they are yet. But let's do a quick check here. If the pH is lower than the pKa, more acidic, more protons, which of these better be more abundant when I get my problem done? HA should be more abundant. So that's a mental check. Right now, when I get to the end, HA better be more abundant or I made a mistake. All right, so I'm going to go ahead and I'm going to subtract 9.5 from both sides, and I'm going to get negative 1.5 over here is equal to the log of A minus over HA. How do I get rid of the log? So not natural log, but the anti-log, essentially. I'm going to take 10 to both sides. So I'm going to take this side to the 10 and this side to 10. Or 10 to this side and 10 to that side. So I can, that's my phone. I can pull out my calculator. And so I'm going to go ahead and do, I'm going to do, well, 1.5, make it negative, and then I'm going to press my anti-log button. You got a log button, probably a shift log, something like that, second log. And what I get out is 0.0316. I'll just keep a bunch of significant figures right now, but really we're not going to worry too much about it. Logs are fun in that you lose significant figures, right? You don't keep all of them. And then taking the log to 10 to the log gets rid of the log, and I end up with that's going to tell me what my ratio of A minus over HA is, right? And I can multiply HA times both sides, and that's equal to A minus. All right, how am I going to solve this? Plug what back in?
The concentration of A minus in terms of HA.
I don't know what A minus concentration is, though, right? So I've got two equations and one unknown. I can't solve it, right? So what I need to do is make up another equation. So what's reasonable? What do I know about if I have HA and I have A minus? What do I know about them in terms of, so this is a percent problem, right? What's the percent abundance of each? If I take HA plus A minus, what percent will I get? 100%, right? So I can make up another equation and say 100% is equal to HA, I'm going to write it as a concentration, but it's not really a concentration, right? Plus A minus. And then this is where I'm going to plug, where I can do a substitution. So now I have an A minus alone here. I have an A minus over there. I can substitute this in for A minus, right? So I can take this and I can put it there because this is equal to A minus. Does that make sense? All right. And so then we're going to do 100% is equal to HA plus 0.03162 HA. Right? How many HA is this? 1. Right? 1 HA plus that. Okay? If you were confused of how many HA this was, don't worry. Lots of people are confused the first time they see it. And so I've got 100% is equal to 1.03162 HA. And so 100% divided by 1.03162 is equal to 97%, 96%, 0.9. So I've got 96.9% of it is HA, which I identified up at the top of the problem, right? So I'm safe. And then if that's the percentage of HA, what's the percentage of A minus? Subtract 100, make the absolute value of it, or subtract it from 100. So 100% minus 96.9%, right? Because A minus plus HA have to make up all of it. And that's going to be, what is it, 4, not 3.1%? 3.1% A minus. So remember back, we said the pH was lower than the pKa, so we should have more of our HA, and in fact we do. So that's a good check there. Before I answer this question, let me just go back and see that my sig figs are like reasonable. You know, I've got 2 sig figs up there, and I've got 2 or 3 down here. Good enough. Okay, yeah.
So I'm confused how we got to HA plus 0.03162 HA.
Okay.
To the one next after that.
How did I get from here?
Yeah, just that whole row has you to the next row.
So what I've got, so you're saying from here to here. Yeah. So what this is you can imagine that this HA is, I could rewrite this HA as 1 times HA. Does that feel better? So now I'm adding, I've got HA and that. Here, let me put another line in between. And so I could rewrite this as HA. I could pull the numbers out into parentheses. 1 plus 0.03162. Does that help? You see how we've got the HA is common, so I could pull it out and have this plus this. And then I just add the 2 together. Okay. Well, maybe talk to me afterwards and we can work on it. Okay. But this is you should always recognize, I'm not usually going to write the 1 here. This is 1 HA. What if this was not such a crazy number? What if this was 1 HA and this was 4 HA? Does that feel better that you add 1 HA plus 4 HA and get 5 HA?
Yeah, okay.
Is that?
Yeah.
Okay, great. Fantastic. Other questions? That was a good question. Remember, if you've got a question, there are like 3 other people in the room at least that have that same question. So make sure you ask them. All right. Let's talk about this problem, how it could be extended a little bit more. So what if instead I'd given you an original concentration there? So what is the concentration of each species, so I'm thinking about each protonation state, of acetaminophen at pH, what do we do, 8? Let's keep the same pH of 8 if the total concentration is, let's say, 55 millimolar. All right. Do you see that you could figure out the percentage and then use the percentage to answer this question? So if I start the problem like that, you could figure out the percentage and do that. Another way you could do it is instead of using, we're just going to use, I'll use the percentage, but I'll quickly highlight another way you could do it. Instead of using this equation, the equation you could make up could be this. And this would actually, our concentration marks would make more sense here. 55 millimolar is equal to the concentration, not in molar. Let me not use it, especially because we're not doing molar here. HA millimolar plus A minus millimolar, right? Instead of that 100%, that would save you having to get the percent and that save you a step. If it's clear how you would do that, feel free to do it in your practice problems. If it's not clear how you would do that, calculate the percent, and then I'll show you how to do this right here. Yes, Bridget.
Since it doesn't say, it just says what is the concentration, can you leave it in millimolar?
Yes. If it doesn't ask for a specific concentration, you can put it in any concentration you want, as long as it's correct. All right, so then how would I solve this? Well, I know my HA is 96.9% of it, so I'm going to turn that into a fraction, 0.6969. I'm going to take that times 55 millimolar equals I'm going to go ahead, to two significant figures, which would be correct here, that's going to be 53 millimolar. Okay. It's 52 or 53.295 on my calculator. And then 50 millimolar, and that was what? My HA or my A minus? HA, right? And I know that because I remember which percent. I can look at the percent, or I remember my pH is lower, and so I should have more of that one. And so that means that 2 millimolar is going to be equal to my A minus. Right? I didn't do that math, but I can. 3,1 times 55 millimolar. I just subtracted from 55. But that's another way. Questions? And maybe I would, since I'm multiplying there, maybe I'd make it two significant figures, or whatever comes out of my calculator. Okay. Which isn't going to be 2.0. I'll put it in my calculator. My calculator, I need to get another calculator. This thing's having seizures today. Here we go. I actually get 1.7. Okay. Questions? Does it bother you they don't add up to 55? It could. If you put down 2, I'd probably just accept it for this one. Okay. But correct significant figures would give you that. Okay, questions on that problem, that process? I'll get some practice worksheets up so people can look at that. I'll get practice worksheets up for both that process and the really quick, is it mostly protonated or mostly not protonated sorts of problems. All right, we're going to move on to chapter 3 then and start talking about DNA and RNA and nucleotides. So we've got 3 nucleotides now, or 4 nucleotides down here. So nucleotides and nucleic acids. What we have down here is we have the ribonucleotides. So there are 4 of them. What we can look at here is, so if you look at in terms of this picture, we've got the pink is going to be just the nucleoside, which is a nucleotide without the phosphate on it. But otherwise, remember, we talked about how you recognize these is you have a nitrogenous base, and it's attached to a ribose sugar, and that's attached to a phosphate. That's how you know you have these. Okay, you're going to need to know the structures and be able to draw the structures. Yes?
What did you say a nucleoside was? A nucleotide without one?
The nucleoside is without the phosphate. So nucleoside is the pink, going with the pink color of the names on the bottom. Okay, and we know these are, how do we, there's 2 ways we know that these are RNA nucleotides. Yes?
You look at the sugar, it's a ribose sugar, not a deoxyribose.
You look at the sugar and you see you have 2 OHs down here. All right, that's going to be important. It's important for, what is RNA? It's important for why RNA makes really weird shapes compared to DNA, which is pretty much a double helix most of the time. And it's important for why RNA degrades faster than DNA. Okay, and we'll talk about all that. So that's one way you can tell. What's the other way? And that's the most important way. What's the other way you can tell? Daniel.
With the U.
Yeah, the U. You're right. You have a U instead of a T. Okay. What I want you to do as well is I want you to be able to recognize these nucleotide names. All right. These are not. We talk about A's, G's, C's, and T's, or we talk about adenines. We'll often talk about the base and things, right? The adenosine, the guanosine, uridine, cytidine, right? But I want you to be able to recognize adenylate, guanylate, uridylate, and citidylate. All right, and I might do those as a mix and match. Yes.
What is the T version of that?
What's the T version of that? We'll look at that in a minute. Okay. Questions on those though? Besides that, we're about to see the DNA nucleotides. Yes.
Wait, so you could be able to recognize what other cardio acid?
I'm asking that you could, if I could do a mix and match with these names. And what I'm trying to avoid is I actually had at one point in my career, I looked and I was reading a paper that was talking about adenylate. I didn't know what it was talking about because I was used to saying adenosine and stuff like that since we used it in graduate school. Okay, so mix and match, you don't have to remember and spell it correctly. All right, otherwise you're going to need to know these structures. I'm not actually going to test them like this. I'll show you. There's 2 ways I'm going to test them, and we'll get to that today. All right, so here we have our DNA nucleotides, the ones that go in DNA. So as Bridget pointed out, these have their deoxy. They're missing a hydroxyl group here, all right, which makes them less reactive. It also reduces the amount of hydrogen bonding you can do, so you can't get as many weird shapes like you do with RNA. RNA can have all sorts of shapes. And then here you need to recognize these names, okay? But it's just the same as the other names, except you put deoxy in front, except for thymine, okay, where we have deoxythymidylate, right? We have thymine here instead. And note the difference between thymine and uracil in terms of the base. Let's see if can I get them both on at the same time? I can if I do this. How are those bases different? Maybe hard to see down here. It's a methyl group, right? The methyl group is the difference there. And there's a good reason for that methyl group. It has to do with DNA repair. We'll go into detail in it in second term. But for DNA to repair, when you have something that's wrong, you need to tell whether it's a normal DNA base or not. And it turns out that there's a way that cytidylate, right? Where cytidine actually will break down to make uracil. It's a very common way in your cells that this becomes this. And then your repair mechanisms don't know. Should it be this? Should it be that? Right? If DNA contained U's, you wouldn't know. So you have to have T's in DNA because your cytidine can't break down to make a T because it doesn't have a methyl group. Does that make sense? You don't need to know that right now, but you'll need to know it second semester if you're still here because you took the class, not because you didn't make it through this one. Okay, questions? All right, so these can go into, we can combine these and make DNA or RNA. And so I want to really quickly point out, you've seen this before, I'm sure, that DNA is like the blueprint of the cell. Okay, so it's like the blueprint for making this building outside of Trexler. There's some fancy blueprint somewhere. They used to be made of paper. Now they're all computerized, right? So you don't really need to worry about them getting torn up. But let's pretend they are still paper, right? You make your fancy blueprint on paper, and you wouldn't want it to get messed up, so you keep it safe. And you make photocopies of it. And that's your RNA. So it's like a photocopy. And these you don't care about so much. You hand them out to all the workers who are building things, and if they get roughed up and broken and stuff, or ripped up, you can always make more, okay? Because you have this safe blueprint, master blueprint you're using. And then the directions that are on those photocopies are used to make proteins. So that's the new science building. May we all live to see it. Actually, it's supposed to be next fall. Yes. Okay, so that's the general path there. All right, this is considered the central dogma of molecular biology. There are other arrows I could draw at this point. Originally, that we thought this was all there was. There are different arrows we can draw around for different ways we can move this. But this is the major path we're talking about. And so it's important that we're able to assemble these things into nucleic acids. Before we do that, I want to just highlight that there are two major categories of these nitrogenous bases. Okay, and those are the pyrimidines and the purines. The pyrimidines are the single ring ones, and the purines are the double ring ones. And you'll see these terms used, especially when you start getting and talking about RNA, which has some funky bases, right? So you get RNA will have bases that are not in the exact form we've talked about for RNA. And there's all sorts of chemistry where you want to talk about purines and pyrimidines. So that's what those are if you hear about those. We may talk about them a little bit. And then the nucleotides come together to make these nucleic acid structures. All right, so notice what's happened here. So we'll have a nucleotide. We're not really going to talk about the mechanism here, and these come together. We have ATPs and GTPs and CTPs that are involved in this sort of thing, NTTPs or UTPs. But you got your basic structure here, right? And what's happened is we've taken a hydroxyl group down here, and we've combined it, or we've linked it to the phosphate of the other group. All right? These structures have direction. I'm actually going to say all of our biological polymers, or all of our major biological polymers, are going to have direction. Meaning they're not the same if you look at them one way versus the other way. For DNA, you've heard of this before probably. We talk about the 5' and 3' ends. All right, so if we look at this structure right here, for both of these, it's not that it's the top, it's the 5' end and the 3' end for both of these. This allows us to talk about the direction, and the direction is super important because it tells you certain things, like all enzymes that would polymerize DNA, right? They're going to make DNA based on a complementary piece of DNA. So they use this as a template, and they'll match it and make DNA over on the other side. But they're all going to go this direction. So when you're making DNA, I haven't talked about it being anti-parallel yet, but when you're making DNA, you always synthesize DNA so that was the other strand being synthesized. But let's imagine this strand's being synthesized. All biological enzymes go this direction. Okay, nobody's found one yet that goes the other direction. I don't know why, but well, I do know why. It's natural selection. But questions? Okay, so we have this direction that's important. So when we think about that direction, I'm going to draw it over here as well. If I have a DNA or an RNA, I've got it's based on the numbering of the sugar. So I've got my base up here. This is going to go down to a phosphate, etc. This is going to go up to another phosphate, or it could be the end. But you can also talk in the middle of it, which is and any of these, what is my 5' side and what is my 3' side, right? So here we're in the middle of one of these polymers, and I'm going to go 1, 2, 3, 4, 5. So that 5' and 3', and these actually for the sugars get primes, which is why we call primes. Okay, but for this thing right here, we call this the 3' end. If we're thinking about right here, that's the 3' direction because that way goes off of this 3' carbon. This is the 5' because it goes off of the 5' carbon. And generally it would be drawn up and down, but I might try to trick you and turn it upside down or something, or sideways. Okay? So I want you to know the structure. Questions? Okay, so there's two questions I'm going to ask, one of two questions that I'm going to ask to test whether you know how to draw these structures or not. And when I say that, the structures of the bases, I'm not going to ask you for the bases all by themselves. I'm going to ask them as part of a polymer. Okay, so what I want you to do, we're going to just practice this right here. All right, I want you to draw me, can you guys see the bases well enough to see the structures? Okay, I want you to draw me a RNA polymer that goes CAT, 5' to 3' CAT. So draw an RNA strand. I'll stretch this out, make sure you can see all the bases. So go and do that, practice that. How do those atoms, how are these, you're gonna have to draw the bases, you don't have the bases memorized yet, but how does it all fit together? And we'll spend, I don't know if we'll spend the whole last 5 minutes on this, but we'll spend a bunch of it. So this is good practice. This is one of the things I might ask. And I always ask one of this or another thing. Yes?
Doesn't RNA use everything?
Oh, it does use you. I'll change that. Depending on the RNA you're looking at, there's so many weird bases that it does actually use T sometimes, but I don't want to be confusing. if you're confused on how something hooks up, I'll try and walk around and kind of look at what people are doing. But feel free to ask. Once you think you have it, feel free to ask me to come over and take a look at it. Especially if you're in the middle and you want me to look at something, because I'm not going to try to navigate the rows without the good motivation. It's not actually real football risks. I'm going to fall on one of you and I'll be okay. But oops, now there's 1, 2, 3. You don't. Chop something on this. All right, you know what I'm going to do? I'm going to start just telling people they can leave when I see something that looks good. All right? Looks good.
It's kind of sloppy, but.
That looks good. Oh, red box.
But otherwise,
Yeah, that's good. Red box. Otherwise,
come up.
red box. Okay, hold on. Yeah. Carbon is between 6 membranes instead of 5 membranes.
Yeah.
You want to practice that. we're out of time. So if you want to go, you can go right now. If you want me to check it, why don't you, I'll check the people on by, but you guys can just go.
That's
If,
It's really nice and fun. We ran out of space.
the same carbon between and here, these phosphorus one. This one needs to be actually up here, actually here,
Yeah.
So you could work on that stuff.
Okay, fine. Yeah.
If you want to work on it, you can check it, or if you want to draw one out.
Okay, yes, I'll do it. Thank you. Mine, I got it.
I don't know that one.
Mine looked exactly like his, except mine bond was the nitrogen, so. I'm gonna, if not, I'm going to tutoring tonight at his.
Oh, you're, okay, yeah. You can
I'll
have
make,
him
I'll
look
make,
at it.
I'll make his.
Have him look at it and make sure it's okay.
Yeah.
So looking at what you've got so far, you've got the right bonds here. You've got that over.
Yeah.
That's it. Mostly I want to look at this part.
And
Yeah,
it's
you
an
too.
it's an OH with the star.
Yeah, this goes. This is not an NH2. It's obviously it's coming off the ring. It's where you got it in the ring. You see that? You got
I just did it.
you got 5 bonds.
I see.
You got nitrogen in the ring, and you got 5 bonds to it.
I see. Yeah.
If you wanted to practice that at some